Good morning, everybody. If you could get settled into your seats. We likely have some people that are waiting live stream, so we want to get started pretty soon. We're just over our 9 o'clock time. There are some seats up front. Don't be shy. Thank you, Jenna. Well, good morning, everybody. Welcome to our 2024 WSS Foundation International Family Conference. Thank you all for being here and from coming far and wide. We always kick off our conference with our why. Why are we here? And we're all here because of some very special kiddos that we all know very, very well. And so I'm going to have your attention go to the screen here. So for those of you that I have not met yet, I'm Libby Woolford. I'm the mom of Francesca, who's a 12-year-old warrior, and this is her decade anniversary. Good morning. Diagnostiversary, I should say. And I wanted to share a story that I share. We do a yearly triathlon, and I shared a story there that I think is kind of appropriate for here as well. And it has to do with a class field trip that I took with Francesca's class earlier this year in February. It was an overnight field trip, probably like you with all of your worries coming here. It was worrying about how she was gonna eat and how we were gonna sleep, and she likes daddy at some points of the day, and that wasn't going to be in place, and then how she was going to do with her friends. And we were only there for about an hour, and there was what is called a gaga pit. I don't know if anybody knows what a gaga pit is, but it is a game where there's a fenced-in pit area, and you have to swat the ball, and if the ball hits you before you swat it, you're out of the pit. And so it starts with all these kids and this ball, and I'm standing there trying to figure out the rules, and Francesca's on the outside of the pit, and then all of a sudden, just on her own, she went in the pit with this mayhem. And slowly, this little boy has his hand on her back, and he's rubbing her back, and I'm watching, and she actually kind of is getting the rules, and they're teaching her, and slowly people get picked out. They're getting kicked out, they're getting hit with the ball before swatting it. And pretty soon everybody's chanting Francesca's name, and everybody is out of the pit, and she's the winner. And for me in that moment, I'm like emotional today, I don't know why. It was knowing that she was supported, and knowing that she was visible by her classmates, and I took this conscious exhale that I realized I had probably been holding my breath since she had entered kindergarten and she was in 5th grade. And so my hope is that while you are here, you look to your left and you look to your right, and with just a line of sight, there's a sign of solidarity. You know exactly what each of you is going through at home. You are behind closed doors, and nobody knows what happens behind those doors. The challenges that you face as you advocate in the doctor's office, in your IEP meetings, with your teachers, within your own family, within your own social group. And I hope that here you can actually take a breath and realize that you are surrounded by this incredible village that is all rooting for you. And you may or may not know it, but there are lifelong friends in this room. There are lifelong cheerleaders for your child in this room. If you are new to this conference, beware, because you will be hooked, and you will have lifelong friends that you'll go on your summer vacations to go and visit. So I hope that just kind of can set the tone for these 2 days, is that there's going to be times that are overwhelming, and there's going to be times that are triggering, but at the end of the day, there's going to be a time where you feel like you can breathe because you're amongst a community that knows, that's walking your same path. So with that, I want to introduce some other cheerleaders that are in your corner and working for your kiddos every day. And that is our board of directors. And I am going to ask them to come up so that everybody knows who they are. But we have Annabelle Corbett, who is joining us from across the pond. And Annabelle is in charge of a lot of she's like our international liaison. And then does a lot with our regional ambassadors, which we are bound and determined to revive that program. And so we will talk about that a little bit more. We have Alex Gillett, who is pregnant and due and with a new little one in January. She just joined us. She has a scientific background, as does her husband. She is our head of research and training. I'm not sure if she knew what she was getting into, but she's handling all of our RareX correspondence at this point, and we can't thank her enough for coming on board. We have Christina O'Keefe. Sorry, I didn't mean to say. Christina has been with the foundation since 2017. Yeah, and we formed in 2016, so almost the whole time. She started out as our treasurer, and as quickly as she could move out of that job, she did. And now she is our head of research and will be falling off the board come March and passing on the torch to Alex. We have Crystal Thurman. Crystal is a godsend. She put together our agenda, as I have shared in our emails to you, worked countless hours to make sure that we had everybody here and a plethora of options for our agenda, and also does a lot with our education and community with our speaker series and outreach. And Thor Misko, I don't know if you can come up or not. Thor is in charge of a lot of our operations. So our to be soon, I hope, unveiled website, our database, our communications, everything that is technical that needs to happen behind the scenes, he takes care of. And I'm missing somebody. Drew. I was like, where is he? He's hiding. Drew is our treasurer. And we have Crystal rolling off our board in March. We have Christina rolling off our board in March. And we have a lot of identified places where we need help. And so we have 2 board positions that are going to be available, along with a lot of infill volunteer needs that we have in order to better support you. We kind of focus everything on 3 buckets: education, community, and research. And we have some holes, and it's really because of bandwidth. We're all parents and all have jobs. So we need more hands and more bodies to fulfill all that we would like to. So as the 2 days progress, if you have any interest or you get inspired and want to get more involved, please talk to one of us. So that's okay. A couple of other people that I wanted to mention, you guys are welcome to stay up here, is, yes. We had a ton of help getting this put together for yesterday and today and tomorrow. Maria Cummings, Abby Wall, Jenna Peccianti, Nina Switsky, Jillian Ambrose, forgetting somebody, Bailey, for putting together the slideshow. Sorry, my phone went off, so I have to go back. And Jillian Algram, she was our on-the-ground recipient of all the boxes over the past couple of months. And then I also wanted to mention Sarah Spear, Kirsten Stewart, Stacy Arnell, and I think that wraps that up. So thank you all You all played an incredible role, and appreciate you. And before I pass it off to Christina here, I wanted to acknowledge a couple of people within our community who are going through some difficult times, and I know that there's quite a few, more than I'm going to talk about, but Sarah Shea, I just said Sarah Spear, Sarah Shea and her daughter Ivy arrived at 3 a.m. last night from Asheville. They just went through Hurricane Helene and have a house that has been flooded, and they had no water and no electricity. They housed their neighbors, they fed their neighbors, they made a 21-hour drive here, and we are all thinking about you and rooting for you, and I can't wait to give you a hug. And then two other families I wanted to briefly mention is at our last conference in Ellicott City, it was very apparent to everybody the relationship that was formed between our youngest attendee and our oldest patient attendee. So Tate was 1 year old when this picture was taken, and Brett was 51 years old, and his mom, Deborah, is pictured there. Brett, we lost about a year ago, so since the last conference, and he passed away of COVID. And Deborah is like our mother hen of all the moms. She's in her 70s and navigated our journey when our journey was a lot more unknown. I can't imagine having navigated that when she did, and all of this time. So I want to send some love to Deborah, and just take a moment to think about Brett, who's incredibly special. And Michelle, the mom of Tate, they were supposed to be here this week, and Michelle's 34 and was diagnosed with stomach cancer about 3 weeks ago and started chemo yesterday. So we just want to keep both of their families in our hearts as we go through these next 2 days, and send them loads of love. So, and on that note, yes, Ned. Today was Brett's birthday, so yes. So thank you for sharing, Ned. All right. I'm gonna bring our research crew up.
But Libby, before you go, I know you usually do this. Who's first-time attendees and?
I do, but you can do that.
You can.
Okay. I was trying to go off.
Oh, we can
what
do
I
it.
normally do.
No, you want to do it together?
Okay. So we usually do everybody stand up.
After 20 minutes, we have to get some
movement.
blood flowing.
So if this is your first conference, and you are brand new to our family conference here in Denver, you're going to take a seat.
Welcome, everyone.
People do always ask this. There are 74 families here. We have United Emirates, we have Czech Republic, we have 3 families from Norway, 1 family from Sweden, 2 families from the UK, researchers from Iceland, and 1 family from Canada. So we have truly become an international group, which is really exciting. And I also did a boys versus girls count, because that also comes up. I think of the families, there were 33 girls and 41 boys. So if Ellicott City was your first conference, you're going to take a seat. If you were present in Sugar Land, Texas, and that was your first conference, you're going to take a seat. And if you were present in Orlando, Florida, and that was your first conference, you're going to take a seat. And then it's the Barnes and the Cummings and us. That we're all in Baltimore at the Sleep Inn and Suites before we knew anything better, and we took a presidential suite. They took the beds out of it, so the headboards were still on the wall, and filled it with chairs, and we did blood draws in the hallway, and we did intakes in our hotel rooms. So we've come a long ways. So thank you.
Yes.
I do usually do that.
I know, it takes a village, right? Thank you, Libby. And a round of applause for Libby. She does an amazing job. Libby, on behalf of the board, and I'm not going to say enough words to make it matter and meaningful for the amount of hours, years that you have put into this foundation. We're always in awe of your ability and your capability and your attention to detail to pull something like this off, and everything you've done for the foundation. So thank you for your leadership. She doesn't want to get off the stage.
I need this.
Yeah. And thank you to Caleb Wall for running to get her coffee, because I think that's what's fueling her today. Well, this is your research crew. We just wanted to walk through a little bit of where we are with research on behalf of the foundation and some of our initiatives. And I'm not going the right way. But I wanted to start off, many of us know what is the Wiedemann-Steiner Syndrome. It was genetically identified in 2012. It's a mutation on a gene that has an important role in our bodies to express other genes, but we're still trying to figure out the mechanics of that. So research is becoming very, very important. And we also are identifying what are all the symptoms from that mutation. And we're recognizing within our community, and you can look at it as you look around this room, WSS is a little different depending on the individual. The spectrum, the severity of symptoms, they're different. And so we are trying to really understand that and build the knowledge for it. So it's really important, the work that we've been trying to focus here at the foundation on research to understand it. With our tremendous network of WSS researcher network as well. So from the beginning with the foundation, we recognized quickly as a kind of rare orphan disease that is WSS, it was really important for us to build those foundational tools, that platform, if you will, to enable and catalyze the momentum for research. And so that has really been the focus for the foundation over the last couple of years. And you can see in each of those boxes across the screen, we really focused in on these different elements that experts have told us are necessary for us to even think about drug discovery, treatments that can help our children. And so first and foremost, early on for the foundation, we focused on animal model. We as a foundation fundraised and we invested $200,000 into creating the first ever WSS mice model, and you'll hear more about that in the next presentation. Very exciting news. So this is definitely an area of tremendous success for us. The next three boxes, though, really are speaking to you, us, patients, our community. As we talk to researchers, you don't understand, and I think we've come to realize more and more, how valuable you are to research. You are needed, your data, your lived experiences are so important for researchers to really understand. So as they're looking at the molecular side, how does that translate into your day-to-day life? Certainly in the clinic practice as well. So at the foundation, we focused in on how can we best gather that data, and one of the things that we partnered with is a patient registry with RareX. We launched that at the conference 2 years ago, and we have really, with the partnership with RareX, encouraged enrollment, and they have a systematic way of taking surveys that's not only on WSS, but across other rare syndromes, which is really important. So if a researcher comes in and says, I want to study something around the GI, it could pull not only our data, but other data, which then we have this more economy of scale approach. So one of the things that we really want to focus in on is how can we encourage more enrollment into these registries, because it demonstrates to researchers, to pharmaceutical companies, we are really interested and we're motivated to help find treatments. So that's one area that we really focused on. The third one is what's called a natural history study. So as WS occurs in an individual from birth to as they age into adulthood, what does that progression look like? And as we've learned going into drug development trials and all of the regulatory processes, it's really important to have these established natural history studies. Those take time. Usually those are longitudinal over someone's life. So one of the partnerships that we have formed is with Citizen Health. They are a company that has a platform to basically pull data from medical records. And so that's a way for us to accelerate research. They will curate that data, and it can be serving as a form of a natural history study, which other rare diseases have been utilizing. So that's something we have launched softly in the last year. We wanted to test it out with the partnership, but we're really focusing on making that open and available to all of you going into 2025. And last but not least is really gathering biosamples and creating cell lines. So as the mutation of WSS occurs in different body symptoms, what is really happening there? So we want to basically create the ability for researchers to study this in those different cells. And so we're really excited today to offer the opportunity for you to contribute biosamples. And so we'll go into that a little bit more. But I just wanted to walk you through kind of these different tools and their importance for us as a patient organization and how we can be a value-add partner to our researchers. And with that, I'll turn it over to Alex Gillett to kind of walk through some of the other initiatives that we've done this year for research.
All right, thank you. As we said earlier, this is my first conference as well, so I'm very excited to be here and actually meet some families for the first time. So what we're currently doing now in 2024, I'm going to talk about briefly. So one of them is looking at the voices of families in research. And if you are connected at all to the foundation, you've probably heard us talking about this burden of illness survey over the past few months. We've really been focusing on this particular survey, which is in the RareX platform, because it's going to be able to inform a lot of decision-making in terms of research and in terms of what you guys are experiencing again in your day-to-day lives, as well as some of these other points I'm going to get to. But that's an important point in our overall research roadmap, which we're in the process of developing as well. So one of the outcomes of this burden of illness survey is investing in research and treatments. So now that we have some data from you guys from the burden of illness survey of what are the most impactful symptoms to you, your families, your children, are to then invest in research on those particular things that are of most importance to you. So we've written a RFP, which prioritizes these most impactful symptoms, which you guys have told us, and we've started looking at opportunities to fund research at $30,000 each, at least to maybe get the ball rolling in a direction and then allow researchers to continue that on that trajectory. The next thing which Christina just mentioned is establishing a WSS biobank. This is one of the things that you can contribute to by giving blood samples today. A biobank is very important for then having a place where researchers can go to have access to these samples for any research they may be inclined to do, and then the footwork is already done for them so that they don't need to do all the work of getting our patients to give more blood samples, which we know is taxing enough to do one time, never mind being asked for multiple studies. And then the last thing, which is again connected to that first point of burden of illness survey, is collaborating with clinicians and Dr. Wendy Jones, who initially identified WSS, to develop clinical health guidelines. This is going to be a very important document for us. This is what you'll be able to take to your doctors and show them this is the best practice right now from this array of experts that are looking at our disease and how to treat X symptom, Y symptom, et cetera, all the way from initial diagnosis to kind of individual symptoms. So we just had an exciting meeting about that last night. The ball is rolling there, but they are looking at using our information that we've given them from the Burden of Illness Survey to incorporate into that as looking at what is important to the community.
And I'll just add, much of these initiatives are well underway. The clinical healthcare guidelines, just to help manage expectations, we're looking at a 2-year time horizon. So we hope for the completion towards the end of 2026. But this is a very important document that we heard loud and clear from many of you, so we were very excited for Dr. Jones and all of the WSS experts globally to come together to really develop this for us. Next, one of our other big achievements this year was bringing on Helen Hernandez. Some of you probably have gotten some emails from her. She's helping to coordinate on site some of these research initiatives. She just came on board 30 days ago. She's really a spark plug, has been just totally driven to help and contribute to this community. And I will say, as a foundation, as a board, we really kind of deliberated and thoughtfully kind of looked at what are our options to keep the momentum and sustain that for research. And with us being working parents, we're volunteers, the capacity was getting limited. So we really thought and have done this investment into Helen as our Chief Scientific Officer. And what we're excited about, I mean, she has accomplished more in the 30 days than I think Alex and I really tried to do the whole year. And so we joke, we're just trying to keep up with her at this point. But I share that because a year from now, I think we're going to see some amazing things continuing coming out of the foundation, and even get beyond just individual family donations, put us in a stronger position to compete for grants through the government or other philanthropic institutions. So I'm really excited to have Helen on board. And with that, I wanted her just to walk through some of the on-site conference research options if you choose to do those. So Helen, welcome.
Thank you, everyone. I am delighted and honored to be here as your new Chief Scientific Officer, and I'm just a really, now I'm a new proponent of Wiedemann-Steiner Syndrome, so I'm here to work for you. I'm here to push the science forward, and I'm delighted to get to meet some of you this weekend. So some of the things we have going on that are actually, here on site are we have the Rare X. The ability to fill out the Burden of Illness survey. At some point I'm going to be sitting with a laptop and you can come find me and I can help you walk through the Burden of Illness survey so that gets incorporated into some of our future research initiatives. And then I also wanted to share that Dr. Hans Bjornson, who's here today, who's right here, thank you, is doing a fingerprint study inspired by some work that he's done in Kabuki syndrome to see if there are differences between the fingerprints of people with WSS compared to those that do not have WSS. And so this is going to be really exciting. If there are differences that we can determine through fingerprints, that would be very helpful, especially in low resource settings, to help people really get to their diagnosis of WSS so that we can meet them and bring them into this community. So thank you for Dr. Bjornson for his work. Thank you. And then Opportunity C is Dr. Rowena Ng and Dr. Jacqueline Harris, and Dr. Harris is right here in the green shirt. And they're going to be doing neurocognitive studies all day today and tomorrow, and that's going to be really important, especially when we try to understand, the features of this disease. And it also ties into the blood draw that's happening. We have a phlebotomist here who's going to draw blood, who's going to look, and then we're going to analyze the blood for lots of different things, including things that are going on at the gene level, as well as the protein level. And then we're going to try to see if there are any correlations between what's happening physically in WSS patients' bodies and their neurocognitive symptoms. So that's going to be really exciting if we can find out more about this disease and how it works and how it impacts our community. So I think that's it, and I am going to pass it back off. Thank you.
Thank you, Helen. So Helen is your resource. If you have a question, contact Helen related to research. If you need something interpreted or to connect with somebody, Helen's your girl. So please utilize her. She's here for you. And then we just want to wrap up. I wanted to then properly introduce our scientific advisory board. These are the experts who help guide us, the board, the foundation, in the research investments that we make. And so I'm really appreciative of all of their expertise. They've been tremendous to us, giving their time and talent to help us kind of explore our path forward for WSS. And with that, we are your research team. Here's our contact information if you have any questions. But again, Helen's your girl if you want to contact her directly. But certainly Alex and I can be a resource for you as well. Thank you all for being here. And I think I'm introducing the next speaker. Or Crystal, you're coming up and introducing the next speaker? Okay. All right. Well, Tinna, if you want to come on up. We got to know Tinna at our conference last year. She works with Dr. Hans Borsen at the University of Iceland, and she is our keeper of our WSS mice. She studies them, she understands kind of what's happening with them to translate into what could happen for us in the clinic, in the doctor's office. And Tinna, you're just a rock star. Can I just say that's your bio? I at least got her to smile and a little embarrassed before your talk, so I apologize for that. But Tinna, I'll let you properly introduce yourself, probably because I can't pronounce some of the Icelandic words. But yes, thank you for being here and sharing your update.
Thank you. Okay. Good morning, everyone. I just wanted to start off, before I go into my session today, I just really wanted to take the opportunity to thank the WSS Foundation for inviting me to come speak today about my research that I've been currently conducting in Iceland with Hans. And before I even get into it, I just feel like I need to get it off my chest. I can just feel the love today from everyone in the family and the community, that it is an absolute pleasure to work with a patient foundation as WSS is. It's incredible to see the families. They're so cohesive, there's so much love, there's so much respect for one another, and this sense of community, as even someone who is not directly affiliated with someone with WSS, it's phenomenal to see. So I just hope that you all are taking it in, and I hope that you really just enjoy the conference and this weekend, because it really is a unique opportunity. So, thank you. So I will be presenting my work today. So as they discussed a little bit earlier, I am doing the mouse work. I am the keeper of the mice, the Wiedemann-Steiner mice. And I took on this project. This has been my PhD project that I recently just wrapped up and graduated. And this has been conducted under Dr. Hans Björnsson, all the way in Iceland. And I'm going to be presenting some of my results today and talking about them, and hopefully give you some insights into what we're doing and how we are trying to really gather as much information as we can using an animal model to see how much we can sort of utilize these information and how we can even, translate them into clinic and stuff. So I'm really excited to get started here today. There we go. All right. So the title of my talk is In-utero Rescue of Neurological Dysfunction in a Mouse Model of Wiedemann-Steiner Syndrome. So even though this may be, so this will be my presentation outline. I'll give you a brief introduction, and then I have sort of two main stories that I want to talk about today. The first is on the creation and characterization of our mouse model. And then my second part will sort of be the testing of the malleability of the neurological dysfunction in vitro and in vivo. So that's sort of the two main talks, and we'll talk all the way through it. So I don't think this really needs an introduction, but for the sake of the talk, Wiedemann-Steiner syndrome is a rare genetic cause of intellectual disability. And as again discussed before, it is characterized by a very broad range of features. Again, like no two cases are identical. It's very broad and dependent on the person. Some of the core feature or maybe some like unifying features include neurological defects like intellectual disability, developmental delay, defects in executive functioning like visual spatial memory. Wiedemann-Steiner individuals often present with distinct facial features, which is another sort of unifying feature, as well as pre- and postnatal growth defects, hypotonia, and hypertrichosis, which is this excessive growth of terminal hair, which is often on the elbows and on the backs, the long eyelashes, the eyebrows. So these are all sort of features that we used to use to identify this disorder before we had any genetic testing. So this is sort of more on the clinical level. So then back in 2012, Dr. Jones discovered that the reason for Wiedemann-Steiner syndrome are defects in the KMT2A gene. So we call these heterozygous de novo variants in the KMT2A gene. And what this means is that we have two copies of KMT2A. We have one on each chromosome. And essentially in Wiedemann-Steiner syndrome, one of those copies is lost. So we have this heterozygous expression or only expression on one of the chromosomes. And these variants often present as de novo or new variants that are not found in the parents. So on a more protein level, this is, has been researched, this protein before. And what we know about this protein, it is a histone methyltransferase 2A, which sounds maybe like a big word, but we know that this is sort of a large protein that's responsible for placing certain marks, which tells the functioning of the cell which genes to activate and which genes to transcribe. So it is sort of a regulator of many aspects. And more specifically, it is known to place mono, di, and trimethylation marks. So it's these specific marks that it places on the chromatin or on the chromosome, and it tells all these regulators where to transcribe, so which genes are supposed to be active and which ones are not. So we're sort of missing this regulation on a cellular level. And what has been researched about KMT2A, so not in the context of Wiedemann-Steiner, but what we know about the functioning of this protein is that it is very important for hematopoietic stem cell regulation, which are your blood cells, as well as being very important for embryonic development. And then in the context of your brain or our nervous system, we know that it's a very important regulator for neuronal proliferation and differentiation, as well as being very essential for postnatal neurogenesis. So we know that it plays a very important role in the brain and in our nervous system, regulating all these stem cells and all these early cells and how they differentiate and how they will behave later on. So this is sort of what we know grossly about KMT2A. So a lot of the work that has been previously conducted has been on similar disorders, or disorders that we term the Mendelian disorders of the epigenetic machinery. And these include Kabuki, which I'm sure a lot of you have already heard about, which is also a sort of similar disorder that has sharing phenotypes. And there's been a lot of work done previously on Kabuki with Hans, with Jackie, and others here. And we're sort of building on the prior knowledge that we know about related disorders, and we're really trying to see whether we can get a gauge of whether Wiedemann-Steiner syndrome sort of behaves in a similar way as these other Mendelian disorders. And these disorders are affecting sort of this epigenetic machinery and this regulation system of which genes are to be transcribed. And we know that Wiedemann-Steiner syndrome is also a part of this sort of umbrella term of disorders that share these features and are sort of defects in the same regulatory systems. And it's very interesting because a lot of these disorders that have the similar genetic component or are defects in a similar system, they share overlapping features such as intellectual disability, some of the growth defects. So we know that there is something very important in the fine-tuning of this sort of vast regulatory network. So we know that Wiedemann-Steiner falls into one of these. So we can really start to build on what we know about the system and what we know about other disorders within the system. And what's maybe most important, what we're really building our research on, is that there has been previous research done on some of these disorders that have shown that some of these have sort of malleability of the postnatal neuronal dysfunction. So we know that, for example, Rubinstein-Taipei, Rett syndrome, Kabuki, very recently Kleefstra syndrome, these are all disorders that share a very similar component. So these are proteins that are highly similar, have highly similar functions. And there's been prior research shown that there's actually some malleability of this network, and these disorders show sort of increased potential of malleability of the neurological dysfunction. And this is really, really important groundwork that we are now building upon. So our sort of overarching question is whether Wiedemann-Steiner syndrome could potentially be yet another treatable cause of intellectual disability. And this is sort of where I come in, because in order to even try to start answering that question, we really need to have the research done in animal models to show that there is room for improvement and there is malleability of the defects, so we can even like start to think about progressing that. So that's sort of where I come in. So this was sort of my introduction, and now I'll be talking about the creation and the characterization of the mouse model. So we created the KMT2A lock-stop-locks mouse model. And in this little schematic, you can see that there are two black lines that show one of each KMT2A genes that we have. And on the top one, we have sort of inserted a break or a stop sign into the mice. And so the gene at the top, one of the two copies that we have, is essentially stopped in the mice. So this is sort of similar to what we actually see in Wiedemann-Steiner syndrome individuals, is that one of the copies that we have is defective or is not working properly. So we've sort of put this stop codon into our mice, and this leads to truncation of the product that is coming from this chromosome or from this gene, which leads to this heterozygous KMT2A expression. So where maybe you and I, or maybe some other people, like from where you would expect there to be 100% KMT2A expression, in a Wiedemann-Steiner individual, this is reduced to around 50%. So one of the copies that we have of this gene is defective. So in this way, we have sort of created a Wiedemann-Steiner syndrome mouse model by inserting the stop codon and sort of mimicking Wiedemann-Steiner syndrome in a mouse. And so the first thing that we did after we had created this mouse model, and Hans very brilliantly designed this, we needed to validate that the mouse model was functioning as it should. So the first thing that we did is that we did some validation tests just to see that the stop break that we put in is both in the place that we needed it to be, as well as it was showing the function that we wanted it to be. So this first here photo shows some sequencing that we did, and we can see that the stop cassette, as we call it, or stop break, is inserted into the right place, which is very good news for us. And then we can also see on this plot where the blue represents the Wiedemann-Steiner mouse model, that it shows around 50% of the relative KMT2A expression. So these information are just essential for us to be to like start working with these models, that we need to know that is the way we designed it. So we did some validation and it was all looking great. So the next thing that we moved on to was characterizing them. So we really, before you can even start thinking about what you can do with this mouse model is that you need to validate that it is a good representation of the syndrome, because you can't really move forward with treating them or seeing if there's any malleability if they don't show any of the symptoms that we see in these individuals. So that was our first step. We needed to validate that the model was correct, and then we needed to validate that it showed some of these symptoms that we see in these individuals. So that was like one of my first steps, and this is absolutely crucial. So now I'm going to go over some of the features that we actually see in our mice. So the first thing that we discussed was, we really wanted to hone in on the neurological dysfunction and see whether the mice would recapitulate some of these symptoms. So one of the first things that we did is that we tested the memory of the mice, and we put them in what we call a Y maze. And here we put the mice in this little Y maze, and we let them explore, and we see how they behave. And what we found is that the lock-stop-lock mice, which are shown here in blue, and this will be a reoccurring theme, so the Wiedemann-Steiner Syndrome mice in blue showed a slight defect in the way they explored the maze. And what this tells us is that, they would sort of re-enter the same arm frequently. They would maybe explore the maze in a systematic way. And what the conclusion that we draw from this is that the mice display some visual spatial defects or defects in their short-term working memory. And this is very similar to what has been seen in individuals. So this was all very reassuring. So then we took a look at the structure of the hippocampus, which is a very important area in the brain, which has been shown to correlate with memory function. And what we found is that on the top here, you can see this V shape in a mouse that does not have the Wiedemann-Steiner Syndrome variant at the top, and the lock-stop-lock model or the Wiedemann-Steiner Syndrome mouse model is at the bottom. And what we found is that when we really quantified and looked at the size and the structure in the mice, we found that the mice had some decreased size of this granule cell layer or this V shape in the brain. And this is actually what has been shown to be very similar in Kabuki syndrome. And they even tested it out and validated it later on in Kabuki syndrome individuals. So we know that this area of the brain is very important for memory function and some of these executive functions. So seeing that this area might be a little bit smaller or have fewer cells in them in the Wiedemann-Steiner Syndrome model could give us sort of a gross area to really see or kind of hone in on an area of the brain where we could do more of our research. So another thing is that we discovered is that we found some craniofacial abnormalities in our mice. And these are CT scanning of our mouse model. So again, at the top, you can see the mice that don't have the Wiedemann-Steiner Syndrome variant, and at the bottom is the Wiedemann-Steiner Syndrome model. And we found some craniofacial structure abnormalities that are similar to the ones that we find in patients. And so this was sort of characterized by an increased width and height of the neurocranium, which is sort of this overarch at the top of the skull. So this is very similar, again, to what we've seen in Kabuki syndrome mouse models, as well as in individuals. Another thing is that these mice display some growth deficiency. So when we just measured their body weight, again, the blue is the lock-stop-lock model, and the gray is their littermates. We find that the mice are smaller, have sort of a decrease in their body weight, which is often prevalent in Wiedemann-Steiner syndrome individuals as well. So this is all confirming what we're seeing in these individuals. Another test that we did was that we looked at their sort of reflexes and muscles. So we found that the mice have sort of indications of hypotonia. So on the first plot, which is further away, that's where we put the mouse pups and we placed them on their backs when they were just 6 days old. And we recorded the time for them to flip over back to their stomachs, which is where they like to be. And we see that this reflex is a little bit delayed in the Wiedemann-Steiner syndrome mice. And this is sort of indicative that they might have some defects in their sort of coordination or trunk control. And then the later test that we did was also just on 6-day-old pups, and we found that we sort of tested their hind limb strength by placing the pups on like a little tube and seeing how long they would hang on for. And we can see that in the third trial, the lock-stop-locks mice would sort of let go sooner. So they would have some hypotonia in their hind limbs. So these are all, again, tests that we really see are taking what we see in these individuals and we're seeing them in the mice, which is great. Another thing that we found, which is maybe a little bit more abstract, is that we found that the mice would have white belly spots. So they would present with some white spotting on their belly. And this was very interesting. So this was not present in all of the Wiedemann-Steiner syndrome mice. This was roughly more than half of them. And this is giving us very important clues to sort of research in general to maybe where the defects are happening, because we know that mice that present with a white belly spot like this have often defects in a certain tissue in embryo development that is sort of giving rise to a lot of these symptoms. And this was very interesting from a research point, even though this is maybe not really comparable to the individuals, but this was really giving us clues to where KMT2A might be working in embryo development. So from a researcher standpoint, this was a really interesting find. So just to conclude my first part, we found, or we created and validated a novel Wiedemann-Steiner syndrome mouse model. We discovered that the mice display some core Wiedemann-Steiner syndrome-like phenotypes, such as the neurological dysfunction, craniofacial abnormalities, growth deficiency, and hypotonia. And these are all essential steps to creating a disease model in mice, is that we really need to be able to validate that the mice have some of these symptoms to be able to assess whether we can alleviate or improve any of these metrics. So this is a very big milestone for Wiedemann-Steiner syndrome. I just really hope that you guys can feel the impact that to have a mouse model that really displays some of these features, this is amazing, and this is all very exciting. So we have sort of created a successful disease model for Wiedemann-Steiner syndrome in mice. So that was sort of the first part of my talk. So the second part that I wanted to discuss was the testing of malleability of the neurological dysfunction in Wiedemann-Steiner syndrome in vitro and in vivo. So the next part to be really be able to start to ask the question of whether we can improve symptom outcomes from these patients, we really need to have sort of biological and animal testing that shows that we can improve this. So Hans designed this model in a brilliant way. So not only does this model serve as a disease model where we can, we have this variant in the mice that replicate Wiedemann-Steiner syndrome in vitro, but we also have a very unique opportunity to remove it. And this is sort of a genetic trick that was designed in a way. So as I talked about earlier, we have put this stop, sort of this break on one of the KMT2A copies that we have. But by putting what we call LOXP sites on either side of it, which are these green triangles, we can use a system that is already in play in some of the bacteria, and we can actually remove it. So we only have this inserted, but then we can remove it. And this creates a very interesting sort of proof of concept method that by crossing this to a nest and Cree mouse model, so here we have another model we are then crossing these two strains together, they express something called the Cree recombinase, which I have sort of put as these scissors. So these mice, the nested mice, they express an enzyme called Cree recombinase, and we sort of depicted them as little scissors because what happens is that when we cross these two strains together, we get pups that have both. So we get pups, and this is sort of what we refer to as our rescue model. So now we have pups that both have these LuxP sites around the brakes and have the Cre enzyme. And what happens is that when we do this, when the Cre recombinase enzyme is expressed or is present in these cells, they actually cut at the LuxP sites. They find the LuxP sites in the genome and they cut right at those sites. So what happens is that when they cut it off is that they remove the stop codon. So when we remove the brake that's causing the Wiedemann-Steiner is that we can actually regain full expression. So before we had around 50% of the KMT2A expression in the mice. After we remove the brake, we're really hoping to see what happens is that when we gain full expression or when we gain this 100% of KMT2A that we expect. So this is a very interesting sort of phenomenon because now we can really assess whether, okay, well, if we regain KMT2A or if we gain it to the expected expression, what happens? Do we see improvements of any of these symptoms that we already have? And the model that we use specifically, the Nestin one, this is sort of happening, this cutting and this regain of KMT2A is happening in the nervous system. So it's not in the entire body, it's just within the nervous system, so the brain and the peripheral nervous system that we regain KMT2A expression, and this is happening around mid-gestation. So what we have created here is a way to test in a proof of concept way what happens if we regain full KMT2A expression around mid-gestation. So that's sort of what the model that we were working with, and I will show you some of the results now. So first, we, of course, needed to validate, if we can remove this stop codon and we can regain full expression, is that really what happens? So in this sort of schematic on the far, you can see that the what we call the lock-stop-lox cassette, or the stop codon, it's not present in mice that don't have it. And then in the middle panel, you can see Wiedemann-Steiner syndrome mice, and you can see that the lock-stop-lox, or the brake, is present both in their brain or within the nervous system, as well as in the tiny little ear clips, so outside the nervous system. But what we then found is that the lanes that are closest to me, you can see that in the ear or outside the nervous system, they still have the brake present, but in the brain or in the nervous system, that is nearly gone. So these are great results that we can really see that the model is working as we predicted and as how we designed it, that within the nervous system or within the brain, this brake is gone. Okay, well now if we've managed to remove the brake from the genome, what is happening on expression level? So again, we looked at the relative KMT2A expression, and we can see that the rescue model, which is depicted here in green, the expression has gone up. So we now know that we have not only removed the brake, but we've also regained expression. So that sort of allowed us to move forward with looking at, okay, well, what happens then? If we can remove it, if we can regain expression in utero, how does that, confer to our metrics that we just sort of described earlier? But before we could even move on to looking at the mice, we needed to know what was happening on a cellular level. So what we did is that we took some cells straight from the nervous system of the mice and we cultured them. And we looked at specifically neuronal progenitor cells because we know that those are really important for Wiedemann-Steiner syndrome pathogenesis. And we looked at sort of what was happening there. So we performed an experiment called RNA sequencing, and these are some of the first results that we have. So as I mentioned earlier, KMT2A is known to be this regulator. It really turns on and turns off genes that are important, and it is this sort of master regulator of a lot of genes that we know are very important. And what we find, and what you can see on this plot, is that we have some genes which are marked in all these little dots that are differentially expressed or are sort of different from the ones that don't have the variant. So here we're comparing the lock-stop-lox model with a mouse model that does not have the variant. And again, all these little dots are the genes, and the ones that are in blue are the ones that are significant. So we can see really a significant difference when we compare the Wiedemann-Steiner model to the ones that don't have the Wiedemann-Steiner variant. And what this is telling us is that a majority of these little dots are on the negative side of the x-axis, which means a lot of the genes that KMT2A is regulating are no longer present. So they're down-regulated in our Wiedemann-Steiner model. And this is sort of what we expected because we know that KMT2A is turning on all these genes and is supposed to be doing that. So when we lose it, we sort of expect that a lot of these genes are now no longer present or are down-regulated. So again, this is very good to see that this sort of is what we expected. So we sort of see a global transcriptional down-regulation of these genes. And so next we wanted to really know, okay, we have all these genes, which are the significant ones in the blue dots, but what are these genes? What are they doing? Why are they important? So we had a look at some of them, and we generated this heat map. And in this plot, you can see the genes that are sort of this violet color are the ones that are downregulated. The ones that are in green or in red, sorry, are the ones that are upregulated. And these are some of the top differential genes. And we see that a lot of them have very important roles in neuronal development, in differentiation and differentiation pathways. So we can really see that the top genes that are dysregulated or maybe not as expected in the Wiedemann-Steiner mouse model, we can really see that that is highly important genes within the nervous system. And what is maybe more important, what maybe is the take-home message from this plot, is that at the top you can see again the different models. The lock-stop-locks model or the Wiedemann-Steiner model is depicted in blue, which is closest to me, and you can see all of the three samples clustered together. They all show the same up and down regulating of these genes, which are on the y-axis. But maybe more interestingly, the green and the gray ones, which are the sort of the baseline or the controls, which are depicted in gray, and the rescue model, they cluster together. So we can see now that when we compare the Wiedemann-Steiner model to the rescue model and the control model, they show a more similar profile. So we're seeing that the effects that we see from the lock-stop-locks or from the Wiedemann-Steiner model is then reversed. So this is sort of the first hints that we could see that some of these transcriptional dysregulation or some of these genes that were used to be turned on and now are turned off, they're going back in a rescue model, which is very convincing that we're really seeing an effect from removing the cassette and regaining KMT2A expression. So these are just some examples of highly important genes, but if we really look at this on a global scale, we find this sort of correlation plot. And on the x-axis here, we're showing the difference between the controls and the Wiedemann-Steiner, while the y-axis is showing the Wiedemann-Steiner variant versus the rescue model. And we find this very striking negative correlation with more than 95% of these top 10 differentially expressed genes following this trend. And what this means, more or less, is that the genes that were downregulated or lost because of the KMT2A variant are now restored in a rescue model. So the rescue model and the controls are showing a very similar pattern. And for a gene that is so important in regulating all of these other genes, seeing that effect restored again is very promising. Another interesting find that we had was that we looked at the specific epigenetic mark that I mentioned earlier, which is this monomethylation signal that KMT2A is responsible for making. So we know that KMT2A is responsible for putting the monomethylation signal in the genome. We know that it's very important to put these monomethylation marks, which again are telling all these other regulators where to transcribe these genes. And we can find that there was actually an increase of the signal when we lost some of the KMT2A from the Wiedemann-Steiner syndrome variant. And this is a little, at a first glance, it might seem a little counterintuitive that, okay, if we're removing KMT2A down to around 50%, why are we seeing this increase? Shouldn't there be a decrease? But more research has shown, for example, in Kabuki syndrome and Kleefstra syndrome, that this is actually the case in a lot of these disorders. And this is really interesting from a research perspective, again, because this is telling us that there might be some sort of compensation mechanism that's happening. So, we're really getting a better insight to what is actually happening within the cell when we lose KMT2A. And maybe more importantly for the context of this talk is that, when we compare the lock-stop-locks model, which again is in blue, and the controls that are in gray, we can see this increase, but again, we see this go down again in our rescue model. So maybe more importantly, we're sort of seeing this restored effects that we see on a cellular level. And another find that we really wanted to see, okay, we see this increase of the signal, but where is this increase in signal happening? We looked at 2 areas of the genome, one for the neuronal progenitor cell enhancers and then neuronal enhancers. And these are areas in the genome that usually have this monomethylation signal that KMT2A puts. And we didn't find any difference in the signal for these early progenitor cells, but what we did find was an increase of this signal in the neuronal enhancers. And what this tells us, based on literature and work that has been done in other disorders, is that we might see a phenotype in these neurons towards a sort of premature differentiation of neurons. And although we haven't explored this further and then seeing this effect, is that this is something that has been widely described, for example, in Kabuki syndrome, that in Kabuki syndrome, neurons, they see a precocious or premature differentiation of the neurons. And that is sort of a disease sort of mechanism that has been described more in Kabuki syndrome. And if we are seeing a similar effect, although we would need to do, of course, more research to see if this is a similar effect that we see in Wiedemann-Steiner syndrome, that could also sort of push us towards a direction to really see what's going on within the nervous system. So this is, again, is sort of hints that we're getting that we are able to take some of the Kabuki research and find similar pathways. Of course, these are distinct disorders, but from a research perspective, sort of looking at certain areas of the brain and certain aspects, we're really able to make sort of an educated guess of in which way to go towards. So this was a very interesting finding. So the last aspect of my talk would be, okay, so now we've seen in the cells, okay, we're seeing this restored gene transcription. We're seeing a restored effect of monomethylation signal. So we are seeing hints on a cellular level when we've just taken the cells and looked at them sort of under a microscope, what's going on in there. What is now happening in our mouse model, in our rescue model that we sort of crossbred, and we can see that there is sort of this increase of KMT2A expression on mid-gestation. And again, this restored KMT2A expression was only happening in the nervous system. So we really wanted to focus on the nervous system metrics that I described earlier and seeing if there are any improved outcomes. And what happens is that when we look at the Y-maze, or the behavior test that was testing the memory that I described earlier, when we compared again the lock-stop-locks mice, or the Wiedemann-Steiner mice, to the controls, we saw this decrease. They showed some visual spatial defects. In our rescue model, we see no defects. We see no changes, or we see no difference between the rescue model and the controls, which is sort of showing us that with a regain of KMT2A expression, we are seeing actually improvements in their memory. And in sort of a similar way, we also see that when we look at the hippocampus structure, which is this V structure within the brain, we find that there is no significant difference between the rescue model and their controls. So we sort of see that this has a restored effect by increasing KMT2A on mid-gestation. So to conclude the second part of my talk, we demonstrated in utero malleability of the neurological dysfunction in a mouse model of Wiedemann-Steiner syndrome. We both did this in vitro or on a cellular level, where we found that the gene expression dysregulation was restored, and we found restored sort of chromatin or monomethylation abnormalities. And then in vivo, for our mice, we both found a restored neurological dysfunction on a behavioral and a structural level. So with all this information that I've now talked about, is that we really believe that sort of the window of opportunity for therapeutic intervention for Wiedemann-Steiner syndrome extends to at least sort of the midpoint of pregnancy, and hopefully sort of expanding that window. So that was pretty much everything that I had to say about that. I would just like to briefly thank Hans Thomas, my supervisor, for bringing me on this project and doing all this incredible work. Of course, I'm not doing this by myself. I have quite the lab that are very supportive. And then again, I cannot thank you guys enough, the families, the individuals. And of course, this would not even been possible without your funding, your fundraising, the importance that you're doing, which directly funded my project. And just while I have the microphone, I would just like to mention, even though I didn't talk about this in this talk, I was one of the scientists that accessed the RareX database for my paper, which is now hopefully going to be published very soon. And that was an incredible opportunity to really work with your data. So the RareX database is incredible, and it had a lot of information, and it is growing and expanding, and is providing us sort of scientists that want to look into this a unique opportunity. So that was really fun to be able to be here at the last conference where this was, just launching, to now a couple years later being actually able to access the data and work with the data. So that was a part of our paper, to sort of give an overview of the syndrome. And I would just like to highly encourage you from a research perspective to consider donating samples and going for the cognitive testing that Jackie is doing, as well as the fingerprint testing that Hans is conducting right now. And as a researcher working with sort of a rare disease, every single individual counts, every single number that we have counts. And it's just very unique to be able to work with a rare disease like this, but to have a community at this level backing it up. You guys are the works of this. You guys are raising the money. You are pushing this forward. So it has been an absolute honor to work on this project, and just all the trust that you guys have given me regarding this project. So I would be happy to take any questions.
Before we go into questions, I'll stop the live stream,